Carbohydrates are synthesized through Kelvin cycle in. Carbohydrates are synthesized through Kelvin cycle in. C3 plants only is wrong. C4 plants only is wrong. C3 and some C4 plants is wrong. All C3 and C4 plants is correct. C3 plants only is wrong. C4 plants only is wrong. All C3 plants and some C4 plants is wrong. Almost all C3 and C4 plants show carbohydrate synthesis through Kelvin cycle, because in C3 plants it directly occurs in mesophyll cells. In C4 plants it occurs in bundle sheath cells present around vascular bundle like a floral wreath. This is called cranch, and that anatomy is called as cranch anatomy. So fourth option. Regeneration of PEP in C4 pathway occurs in. Regeneration of PEP in C4 pathway. Regeneration of phosphoenol pyruvic acid in C4 pathway or Hatch-Lock pathway or beta carboxylation cycle or dicarboxylic acid cycle occurs in. Cytosol of mesophyll cells is wrong. Chloroplast of mesophyll cells is correct. Cytosol of bundle sheath cells is known for glucose synthesis. Cytosol of mesophyll cells is known for carboxylation of phosphoenol pyruvic acid. Chloroplast of bundle sheath cells is known for oxidative decarboxylation of malic acid and occurrence of C3 cycle by carbon dioxide absorbed by RUBP. So, chloroplast of mesophyll cells is the place where regeneration of PEP in C4 pathway occurs. Second option. The correct sequence of the following reactions in C4 cycle. The correct sequence of the following reactions in C4 cycle: phosphorylation, carboxylation, reduction, oxidative decarboxylation, first carboxylation, then reduction, PEP converted into oxaloacetic acid is carboxylation. Conversion of oxaloacetic acid into malic acid is reduction. Then oxidative decarboxylation. Oxidative decarboxylation is malic acid converting into pyruvic acid in bundle sheath cells. Finally, phosphorylation. How? Again, in the mesophyll cells of chloroplasts of mesophyll cells, pyruvic acid takes 2 ATP and converts into phosphoenol pyruvic acid. That is phosphorylation. So, correct sequence is carboxylation, reduction, oxidative decarboxylation and phosphorylation. 2, 3, 4, 1. First option. Wrong match with reference to their site of formation of intermediate compounds in C4 cycle. Wrong match with reference to their site of formation of intermediate compounds of C4. Malic acid, chloroplast of mesophyll cells is perfectly correct. Pyruvic acid, chloroplast of bundle sheath cells is perfectly correct. Oxaloacetic acid, cytosol of mesophyll cells is correct. Cytosol of mesophyll cells is the place where oxaloacetic acid is formed. Phosphoenol pyruvic acid, cytosol of mesophyll cells is wrong. It is formed in the chloroplast of mesophyll cells and then transferred to cytosol of mesophyll cells. So, wrong match with reference to the site of formation of intermediate compound in C4 cycle is phosphoenol pyruvic acid is not formed in cytosol of mesophyll cells. It is formed in the chloroplast of mesophyll cells, then transferred to the cytosol of mesophyll cells. Whereas malic acid is formed in the chloroplast of mesophyll cells, pyruvic acid is formed in the chloroplast of bundle sheath cells, oxaloacetic acid is formed in the cytosol of mesophyll cells, but phosphoenol pyruvic acid is formed in the chloroplast of mesophyll cells, not in the cytosol of mesophyll cells. So, fourth option. Assimilatory power required to reduce or required to produce one glucose molecule through C4 pathway. Assimilatory power required to produce one glucose molecule through C4 pathway is 18 ATP, 12 NADPH2 is wrong. 30 ATP, 18 NADPH2 is wrong. 18 ATP and 12 NADPH2 is wrong. The correct answer is 30 ATP and 12 NADPH2. Why? Because for 1 CO2, 5 ATP and 2 NADPH2. For 6 CO2 to produce glucose, it is 6 into 5, 30 ATP, 6 into 2, 12 NADPH2. So, assimilatory power required to produce one glucose molecule through C4 pathway is 30 ATP and 12 NADPH2. Assimilatory power consumed in mesophyll cell chloroplast of C4 plants for the fixation of each CO2. Assimilatory power consumed in the mesophyll chloroplast of C4 plants for the fixation of each CO2 is 5 ATP and 2 NADP H2. Second option. Assimilatory power utilized in excess in C4 plants for the synthesis of one glucose molecule. 12 ADP, 6 NADP H2 is wrong. 5 ATP, 2 NADP H2 is wrong. 12 ATP, 12 NADP H2 is wrong. 12 ATP is correct. This is excess. 18 in C3 plants, 30 in C4 plants. So excess is 12 ATP. Which of the following is low in C4 plants and high in C3 plants? Efficiency of CO2 utilization, requirement of assimilatory power for glucose synthesis, photosynthetic yield, all cannot be compared, but CO2 compensation point. The CO2 compensation point in C3 plants is around 50, in C4 plants is 0. What is CO2 compensation point? The concentration of carbon dioxide in atmosphere at which the concentration of carbon dioxide in the atmosphere at which the loss of CO2 by respiration is compensated by the fixation of CO2 by photosynthesis is called carbon dioxide compensation point. The loss of CO2 by respiration is compensated by the fixation of CO2 by photosynthesis is called as carbon dioxide compensation point. This is very low in C4 plants than in C3 plants. C3 plants it is very high, almost 50. In C4 plants it is 0. Wrong match of the following. Wrong match of the following. Hill, evolution of oxygen from water, correct. Emerson, discovery of two pigment systems, correct. Warburg, inhibitory effect of oxygen on photosynthesis, this is very important. Effect of oxygen on the rate of photosynthesis is popularly called Warburg's effect. Liebig, Liebig is known for law of minimum. Blackman is known for law of limiting factors. Law of limiting factors. So Liebig is not for law of limiting factors. Liebig is known for law of minimum, based on which Blackman proposed law of limiting factors. So wrong match in the following is Hill, evolution of oxygen from water is true. Emerson, discovery of two pigment systems by Emerson's enhancement effect and proposing a concept called red drop. Warburg, effect of oxygen on the rate of photosynthesis or inhibitory effect of oxygen on the rate of photosynthesis is correct. Liebig, law of limiting factor is wrong. Liebig is law of minimum, not law of limiting factors. Rubisco also acts as, Rubisco not only acts as carboxylase, it is also acts as oxygenase. Why? Because Rubisco expansion is RUBP, carboxylase and oxygenase. When carbon dioxide is more than oxygen, it functions like carboxylase and helps in the occurrence of C3 cycle. When oxygen is more than carbon dioxide, it functions like RUBP oxygenase and leads to the occurrence of photorespiration. What is photorespiration? Even plant starts liberating carbon dioxide in the presence of sunlight is photorespiration. So it also acts as oxygenase. This is assertion reasoning. Carbon dioxide compensation point is high for C3 plants. Definitely carbon dioxide compensation point is high for C3 plants. C3 plants cannot utilize carbon dioxide as its low level in at its lower level in atmosphere. So C3 plants cannot utilize CO2 at their lower level in atmosphere because their carbon dioxide compensation point is very, very high. So both A and R are correct. R is the correct explanation of A. Another assertion reasoning. Etiolated plants show poor growth of stem. What is etiolation? When a plant is grown in darkness, the plant will become pale in color, tall in nature, and leaves are very much stunted, very short. Etiolation of plants can be observed under complete dark conditions. Etiolated plants show poor growth of stem is wrong. Etiolated plants show maximum growth of stem. Etiolation of plants can be observed under complete dark condition is correct. So etiolated plants show poor growth of stem is wrong. Etiolated plants always show maximum growth of the stem. So A is false or is true. Fourth option. The limiting factor of photosynthesis is usually. The limiting factor of photosynthesis is usually carbon dioxide. Blackman proved this. Whenever this is light intensity, and here it took rate of photosynthesis. Whenever all the factors are perfectly present, photosynthesis occurred from A to B. At one point of time, photosynthesis stopped for a while from B to C because CO2 is less. Then what Blackman has done? Proposed or given carbon dioxide. Again photosynthesis occurred from B to D. Again it is stopped from D to D because CO2 is more. So whenever CO2 is less, whenever CO2 is more, the rate of photosynthesis completely minimized. Any external factor that shows maximum effect on the rate of photosynthesis, any external factor that shows maximum effect on the rate of photosynthesis, even in small amounts or larger amounts, is called limiting factor. So Blackman proposed this concept. What is limiting factor? Any photosynthesis is controlled by many external factors. Photosynthesis is controlled by many external factors. Any external factor that shows maximum effect either in very less amounts or in very huge amounts, then it is called limiting factor. So the limiting factor of photosynthesis is carbon dioxide. Why? When carbon dioxide is less, when all the cofactors or all the factors are available, photosynthesis will become zero. When carbon dioxide is high, then also photosynthesis becomes stops. So the limiting factor of photosynthesis is usually carbon dioxide. C4 plants are different from C3 plants with reference to type of end product of photosynthesis, both is same, glucose. Number of NADPH2 that are consumed during preparation of sugar is same, 12 NADPH2. Type of pigments involved in photosynthesis is same, chlorophyll A, chlorophyll B. Substance that accepts CO2 in carbon assimilation is different. CO2 substance that accepts CO2 in carbon assimilation in C4 plants is PEP, phosphoenol pyruvic acid. Substance that accepts CO2 for carbon assimilation in C3 plants is RUBP, ribulose 1,5-biphos. Here the basic difference lies between C4 plants and C3 plants. So C4 plants are different from C3 plants with reference to the substance that accepts CO2 in carbon assimilation. First option. Which of the following would not be a limiting factor in photosynthesis? Limiting factor of photosynthesis, CO2 will be a limiting factor definitely. Light is also a limiting factor definitely. Chlorophyll is also a limiting factor because light, CO2, chlorophyll all directly involved in photosynthesis. Oxygen is a byproduct of photosynthesis because whenever glucose or whenever carbon dioxide combines with water, glucose is formed, water is liberated and oxygen is liberated. So as oxygen is a byproduct, it will not be a limiting factor. The effect of oxygen comes only when oxygen concentration is more and carbon dioxide concentration is less, then photorespiration takes place. That is called effect of oxygen on the rate of photosynthesis, known as Warburg's effect. So which of the following would not be a limiting factor in photosynthesis? Definitely oxygen. This is a 3-column matching. Study the following table with reference to photorespiration and correct combinations are: glyoxylate formed in peroxisomes enters to mitochondria, wrong, because what enters mitochondria is glycine. Hydroxypyruvate formed in peroxisomes enters chloroplast is wrong, because hydroxypyruvate remains in the peroxisomes, become glyceric acid, then enters chloroplast. Phosphoglyceric acid forms in peroxisomes and enters to chloroplast is wrong. Phosphoglyceric acid is formed in the chloroplast only. From glyceric acid by utilization of ATP, what is formed? Phosphoglyceric acid is formed. Here peroxisome is not at all involved. Glycolic acid is formed in chloroplast and enters peroxisomes is correct. This is the first step where RUBP instead of taking carbon dioxide takes oxygen, becomes one molecule of 3-phosphoglyceric acid and one molecule of phosphoglycolic acid. Phosphoglycolic acid undergoes dephosphorylation and forms glycolic acid. Glycolic acid moves from chloroplast to peroxisome. So in these combinations, the only correct combination is glycolate. Site of synthesis is chloroplast. After the site of synthesis in chloroplast, it is enters into peroxisomes. Fourth option. This is an assertion reasoning. Photorespiration is also called photosynthetic carbon oxidation cycle. Photorespiration is also called photosynthetic carbon oxidation cycle is correct. In photorespiration, the total photosynthetically fixed carbon is transferred to phosphoglycolic acid. In photorespiration, the total photosynthetically fixed carbon is transferred to phosphoglycolic acid is wrong, because one molecule of 3-phosphoglyceric acid is formed which involves in dark reaction. One molecule of phosphoglycolic acid is formed which is transferred as phosphoglycolate. So A is true, R is false. Third option. Cell organelles involved in glycolate metabolism. Glycolate metabolism is nothing but photorespiration. Glycolate metabolism is nothing but photorespiration. The cell organelles involved in photorespiration are C2 cycle are chloroplast, peroxisomes, mitochondria. So glyoxysomes is wrong. What is a glyoxysome? It is discovered by Bredenbach, found in all cells. It helps in the conversion of fats into carbohydrates, which is known as glyoxylate cycle. Chloroplast is correct. Endoplasmic reticulum is not at all involved here. Peroxisomes is correct. Lysosomes are not involved. Mitochondria is correct. So answer is B, D, F. Chloroplast, peroxisomes, mitochondria. So cell organelle involved in glycolate metabolism are chloroplast, peroxisomes, mitochondria. B, D, F. Third option. Identify the incorrect combination with regard to photorespiration. Incorrect combination. Dephosphorylation of phosphoglycolate occurs in the chloroplast is correct. Because only dephosphorylation of phosphoglycolate, what is formed? Glycolic acid is formed, it occurs in chloroplast. Oxidation of glycolate occurs in peroxisomes, definitely correct. Oxidation of glycolic acid converts into glyoxylic acid. Hydrogen peroxide is formed which is broken down into H2 and O2 in peroxisomes is correct. Amination of glyoxylate occurs in mitochondria is wrong. Amination of glyoxylate occurs in peroxisomes itself. Decarboxylation of glycine occurs in mitochondria is correct. 2 molecules of glycine enter mitochondria, liberates CO2 and forms serine. Everything occurs in mitochondria. So amination of glyoxylate in mitochondria is wrong. Amination of glyoxylate occurs in peroxisomes only. So third option. Arrange the following compounds in ascending order based on the number of carbon atoms in them. Based on the number of carbon atoms in them. Glycolate, glycolic acid 2 carbon. Serine 3 carbon. Erythrose 4 carbon. RUBP 5 carbon. Carotene 40 carbon. So in ascending order every time carotene will be the last. Like that we can identify this. If 4 is the last, that will be the answer. So 1 glycolate 3, 2 serine, glycolate 2, serine 3, erythrose 4, RUBP 5 and carotene 4. 1, 2, 3, 5, 4. So based on the number of carbon atoms present in them, ascending order is glycolate 2 carbon, serine 3 carbon, erythrose 4 carbon, RUBP 5 carbon, carotene 40 carbon. So answer is 1, 2, 3, 5 and 4. Third option. Which of the following occurs in the peroxisomes during C2 cycle? Peroxisomes during C2 cycle means peroxisomes during photorespiration. Formation of glycolate, it is occurring in chloroplast. Formation of glycine occurs in peroxisomes. Formation of serine, not occurs here. Formation of serine occurs in mitochondria. Formation of glycerate is occurring in peroxisome. So answer is 2 and 4, second option. During photorespiration, both 2 carbon and 3 carbon compounds are produced in the 2 carbon compound is glycolic acid, 3 carbon compound is serine. So where they are produced means chloroplasts and peroxisomes. During photorespiration, both 2 carbon and 3 carbon compounds are formed in chloroplasts and peroxisomes 1 and 2. First option. Identify the correct sequence of events in light reaction. Excitation of P700, energy transfer to P700, oxidation of P700, absorption of light by light harvesting complex. Fourth one first, absorption of light by light harvesting complex. Energy transfer to P700 is second. Excitation of P700 is next. Oxidation of P700 is last. So 4, 2, 1, 3. 4, 2, 1, 3 is the combination. Find the correct sequence of cell organelles involved in photorespiration. So photorespiration, chloroplasts, peroxisomes, mitochondria. RUBP, 5 carbon compound, instead of taking carbon dioxide takes oxygen. In the presence of RUBP, oxygenase forms 1 molecule of 3-phosphoglyceric acid, 1 molecule of phosphoglycolic acid. This phosphoglycolic acid undergoes dephosphorylation and forms glycolic acid. Glycolic acid enters here and converts into glyoxylic acid. Glyoxylic acid becomes glycine. Here H2O2 is formed, which is broken down into H2O and half O2. Here deamination takes place. 2 molecules of glycine enter here, liberates carbon dioxide and forms serine. Serine comes back here, becomes hydroxypyruvate by deamination. Hydroxypyruvate becomes glyceric acid. Glyceric acid enters here, converts into phosphoglyceric acid, which regenerates RUBP. So this is the actual C2 cycle or photorespiration. So find the correct sequence of cell organelles involved in photorespiration. Answer is 2, 1, 3. First, double unit membrane cell organelle involved in photophosphorylation, chloroplast. Double unit membrane cell organelle involved in photophosphorylation, chloroplasts. Then 1, single unit membrane microcell organelle, peroxisomes. Peroxisomes are surrounded by single unit membrane, that is peroxisome. Single unit membrane compounds involved in a single unit membrane microcell organelle, microbodies. Double unit membrane cell organelle involved in oxidative phosphorylation, that is mitochondria. So first, double unit membrane cell organelle involved in photophosphorylation is chloroplast. Then single unit membrane microcell organelle is peroxisomes 1. Then double unit membrane cell organelles involved in oxidative phosphorylation is mitochondria. So find the correct sequence of cell organelle involved in photorespiration are 2, 1, 3. First chloroplast, then peroxisomes, then mitochondria. Double unit membrane cell organelle involved in photophosphorylation is chloroplast. Single unit membrane microcell organelle, peroxisomes. Double unit membrane cell organelle involved in oxidative phosphorylation is mitochondria. 2, 1, 3. Which is the following is true about photosystem 2? Which is the following is true about photosystem 2? It begins the non-cyclic pathway of ATP synthesis, correct. Definitely PS2 takes light harvesting complex light, get excited, takes 2 electrons which were taken by pheophytin, from pheophytin to quinone cycle, from quinone cycle to cytochrome B6F, from cytochrome F to plastocyanin to PS1, finally to ferredoxin, where ferredoxin gives electrons and resulting in the reduction of NADP to NADPH2. So it begins the non-cyclic pathway, and excited PS2 gets 2 electrons by the photolysis of water in the lumen side of thylakoid in the presence of 4 molecules of calcium, manganese, chlorine. Whenever one water molecule undergoes photolysis of water, 2 protons accumulate in lumen. After the quinone cycle, 4 protons come from stroma matrix to lumen with the help of plastosemiquinone. So name the pigment which transfers the protons from stroma matrix to lumen side of thylakoid is plastosemiquinone. So it begins the non-cyclic pathway of ATP synthesis is correct. It has P680 as its reaction center. So PS2's reaction center is always P680. It replaces its lost electrons through photolysis of water. Definitely whenever it loses electrons, it is in excited state. To bring it back to the ground state, what happens? Water molecule breaks up, and this water molecule provides 2 electrons to bring the PS2 to the ground state. It is the site of NADPH formation is wrong. The site of NADPH formation is stroma matrix. How? All the electrons liberated from water through non-cyclic photophosphorylation go to PS1. From PS1 they go to unknown compound, from unknown compound to first and last electron acceptor and donor, that is ferredoxin gives electrons to NADP plus and converts it into NADPH2. So it is the site of NADPH formation is wrong, whereas it begins the non-cyclic pathway of ATP synthesis is correct. It has P680 as its reaction center is correct. It replaces the lost electrons through photolysis of water is correct. It is the site of synthesis or site of NADPH formation is wrong. So except D all are correct. Except A all are correct is wrong. A and B only correct is wrong. Except B all are correct is wrong. Except D all the remaining 3 are correct. This is an assertion reasoning. Photorespiration is not entirely a wasteful process. Photorespiration is not entirely a wasteful process. This pathway recovers 75% of carbon to the PCR cycle and also known to protect C3 plants from photooxidative damage. Photorespiration is not entirely wasteful process. Why? Because photorespiration recovers 75% of the carbon to PCR cycle again. So when continuous carbon dioxide is taken from atmosphere by the plants to perform photosynthesis C3 cycle and produce glucose, definitely the percentage of carbon dioxide in atmosphere reduces. In order to regain that carbon dioxide, photorespiration must occur. So photorespiration is not entirely a wasteful process is correct. A sort of carbon dioxide to the atmosphere is given by photorespiration also. This pathway recovers 75% of carbon to the PCR cycle is correct, and also known to protect C3 plants from photooxidative damage. Photooxidation means the destruction of chloroplast at higher light intensities. At higher light intensities. This is called photooxidation. This photooxidation effect on chloroplast is minimized by carotenoids, that is carotins and xanthophylls. That's why carotenoids are popularly called shield pigments. They are popularly called as shield pigments. So destruction of chloroplast in the presence of high light intensities is called as photooxidation effect, which is nothing but photorespiration. And photorespiration is not entirely a wasteful process is true. This pathway recovers 75% of the carbon to the PCR cycle and also known to protect C3 plants from the dangerous effect of photooxidative damage. So A and R are correct. R is the correct explanation of A. Match the following with reference to light phase. Number of photons required for the evolution of one oxygen molecule. Number of water molecules photolyzed for the evolution of one oxygen. Number of electrons transported from evolution of one oxygen molecule. Total number of protons released into the lumen after the evolution of one oxygen molecule. A2. Number of photons required for the evolution of one oxygen molecule, because for 2 electrons 4 photons, for 4 electrons 8 photons, for 2 electrons only half O2 will come, for 4 electrons full O2 will come. Then B4, number of water photolyzed for the evolution of one oxygen molecule is 2. Number of water molecules photolyzed are 2. For one water molecule, half O2. For full oxygen molecule, 2 water molecules. Number of electrons transported per evolution of one oxygen molecule, 4. Number of electrons transported for 2 electrons, half O2. For 4 electrons, full O2. The total number of protons released into the lumen for the evolution of one oxygen molecule. The total number of protons released into the lumen for the evolution of one oxygen molecule. Whenever one quinone cycle takes place, one water molecule is broken, 4 half O2 are liberated and 6 photons get accumulated, 2 from the water and 4 from the stroma matrix through plastose, semiquinone and quinone cycle. For half O2 it is 6 protons, for full O2 it is 12 protons. So, answer A2, B4, C5, D3. Second option will be the answer for this. 4 pyrrole rings are present in these pigments. 4 pyrrole rings are present in these pigments means carotenes does not show any pyrrole rings. Phycoerythrin is a phycobilin. Pyrrole rings are present but they are straightened. They are not formed in the form of a group. Whereas in chlorophyll, 4 pyrrole rings are straightened. 1, 2, 3, 4. All are connected and at the center they show a magnesium, and to the 4th pyrrole ring a 20 carbon alcohol chain phytol tail is attached. So, chlorophyll contains 4 pyrrole rings arranged in the form of a cyclic manner in the case of chlorophyll, whereas in phycobilins pyrrole rings are present but they are not in the form of a cyclic manner, they are arranged in a straightened manner. So, 4 pyrrole rings are present in these pigments means not in carotene. Carotene is C40H50, it is a straightened carbon. Phycoerythrin, which is nothing but phycobilin, and chlorophyll 2 and 3. The functions of carotenoids in photosynthesis: absorbing light energy and transferring to reaction center. That's why it is popularly called accessory pigment. Absorbing light energy and transferring it to reaction center is done by both phycobilins and carotenoids. So, they are accessory pigments. Protecting the chlorophyll from photooxidation. Protecting the chlorophyll from photooxidation, that's why these are also called shield pigments. Protecting the enzymes of stroma from thermal oxidations. Carotenoids does not involve in this type. Protecting the enzymes of stroma from thermal oxidation is wrong. So, absorbing light energy and transferring to reaction center is correct. Protecting chlorophylls from photooxidation, that's why they are popularly called shield pigments, is correct. So, answer is 1 and 2, third is not involved. So, 4. This is an assertion reasoning. Thylakoid membrane show photophosphorylations. Thylakoid membrane shows photophosphorylations. Why? Photosphorylation means formation of ATP. It has PS1 and PS2. PS1, PS2 helps in electron transport. Whenever electron transport takes place, proton translocation occurs. Whenever proton translocation occurs, ATP formation occurs, that is photophosphorylation. But completely not because of PS1 and PS2 ATP formation. There is another complex called coupling factor complex or ATP synthase, by which ATP formation occurs. So, thylakoid membrane show photophosphorylation is correct. It has PS1 and PS2 is correct, but both are not linked. That's why A and R are correct. R is not the correct explanation of A. Second option. Another assertion reasoning. Reaction center is need to convert light energy into chemical energy. The main function of reaction center of pigment systems, whenever light harvesting complex absorbs sunlight, it transfers to the reaction center by a special method called resonance transfer or inductive resonance. So, reaction center is need to convert light energy into chemical energy is correct. All pigment molecules can convert light energy into chemical energy is wrong, because Carotenoids and phycobilins are pigment molecules which do not have any reaction center. They only trap the light energy and transfer it to reaction center. So, reaction center is need to convert light energy into chemical energy is correct. All the pigment molecules can convert light energy into chemical energy is wrong. So, A is true, R is false. Third option. 70, that is assertion. PS1 and PS2 are structurally similar. Definitely both are structurally similar because both have light harvesting complex and reaction center. Both trap the light energy from light harvesting complex and send it to the reaction center by inductive resonance or resonance transfer. Both are involved in trapping light energy. Both are involved in trapping light energy is true, but next what happens is not given. So, both A and R are correct. R is not the correct explanation of A. Another assertion reasoning. For this photosynthesis, assertion reasoning, matching are very important. That's why so many questions are given regarding matching and assertion reasonings. Chlorophyll molecule has two parts known as porphyrin and phytoltyl. So, chlorophyll definitely contain two parts known as porphyrin and phytoltyl. All chlorophylls have similar molecular structure is wrong. Chlorophyll A has some components, chlorophyll B has some components, C, D, A all show different. All chlorophylls have similar molecular structure is also same. Chlorophyll has two parts, that is porphyrin head and phytoltyl is correct, but both are not linked together. So, A and R are correct. R is not the correct explanation of A. Arrange the following substances in a sequence during dark reaction in chlorella pyrenoidosa. How they are formed? Erythrose monophosphate, phosphoglyceraldehyde, xylose monophosphate, ribose monophosphate. Always first formed is phosphoglyceraldehyde. Second one. Always first formed is phosphoglyceraldehyde. 6 molecules of phosphoglyceraldehyde are formed. 1/6 goes to the lumen. 5/6 remains in stroma matrix. 1/6 in lumen produces glucose. 5/6 in stroma matrix regenerates RUBP. So, phosphoglyceraldehyde. Then erythrose monophosphate. Erythrose monophosphate. Then xylose monophosphate. Xylose monophosphate forms 2 times. Erythrose monophosphate and xylose monophosphate. Finally, ribose monophosphate. So, answer is 2, 1, 3, 4. Phosphoglyceraldehyde, erythrose monophosphate, xylose monophosphate, and finally ribose monophosphate. Fourth option. Arrange the following formation of following substance in C4 cycle in a sequence. C4 cycle in a sequence. Phosphoenol pyruvic acid, malic acid, oxaloacetic acid, pyruvic acid. Phosphoenol pyruvic acid is not formed first. Phosphoenol pyruvic acid is first compound which undergoes carboxylation. So, first formed is always oxaloacetic acid. First formed is always oxaloacetic acid. This oxaloacetic acid is formed in the lumen of mesophyll cells by carboxylation of phosphoenol pyruvic acid. So, first formed compound oxaloacetic acid, that is why this cycle itself is called as C4 cycle. Next, malic acid. How? The oxaloacetic acid moves to bundle sheath cells. Oxaloacetic acid moves from lumen to mesophyll cells of chloroplast of mesophyll cells, where they undergo reduction and produce malic acid. So, malic acid second form. Then malic acid goes to bundle sheath cells, undergoes oxidative decarboxylation to produce liberate CO2 and NADPH2 to produce pyruvic acid. So, third compound formed is pyruvic acid. Pyruvic acid now moves from bundle sheaths to mesophyll cells, where in the presence of pyruvate dikinase they utilize 2-ATP, undergo phosphorylation and produce phosphoenol pyruvic acid. So, first formed is oxaloacetic acid, next formed is malic acid, then formed is pyruvic acid. So, if you write in a sequence, oxaloacetic acid, malic acid, pyruvic acid and phosphoenol pyruvic acid, which again undergoes carboxylation and repeat the cycle. So, sequence is 1, 2, 3, 4. So, oxaloacetic acid, malic acid, pyruvic acid, phosphoenol pyruvic acid. First option 3, 2, 4, 1. This is the 3 column: enzyme, substrate form, substrate involved, product form. Trans-ketolase 3-phosphoglyceraldehyde, substrate is correct, but fructose biphosphate is wrong. Here xylulose 5-phosphate and erythrose 5-phosphate are formed. Trans-androlase 3-phosphoglyceraldehyde, enzyme is correct, substrate is correct, but ribose monophosphate is wrong. 3-phosphoglyceraldehyde trans-aldolase means it must be either fructose 1,6 biphosphate or pseudoheptose 1,7 biphosphate. Epimerase xylulose monophosphate, ribulose monophosphate is correct. Whenever xylulose monophosphate undergoes epimerization in the presence of epimerase, definitely ribulose monophosphate is formed. So, third is correct. Aldolase 3-phosphoglyceraldehyde, aldolase 3-phosphoglyceraldehyde, fructose bisphosphate. Whenever aldolase is present, 3-phosphoglyceraldehyde combines with dihydroxyacetone phosphate, resulting in the formation of fructose 1,6 biphosphate. So, trans-ketolase 3-phosphoglyceraldehyde, fructose bisphosphate is wrong. Trans-aldolase 3-phosphoglyceraldehyde, ribulose monophosphate. There in the first one, either xylulose or erythrose has to come, they have not come. Next in the one, pseudoheptose 1,7 biphosphate has to come, didn't come. But epimerase, xylulose monophosphate epimerization produces ribulose monophosphate is very much correct. Aldolase 3-phosphoglyceraldehyde combining with dihydroxyacetone phosphate produces fructose biphosphate is correct. So, answer in this combination, 2 correct combinations: enzyme, substrate involved, product formed is 3 and 4. Another 3 column: stage of photosynthesis, feature 1, feature 2. Non-cyclic electron transport, NADPH are formed, oxygen is evolved, perfectly correct. Perfectly correct. Non-cyclic electron transport, NADPH2 are formed, oxygen is evolved is correct. Cyclic electron transport, NADH2 is formed, oxygen is not evolved is wrong. In cyclic electron transport, no question of liberation of NADH2. Only oxygen is not evolved is also correct. Oxygen is not evolved is also correct. Cyclic photophosphorylation, NADH2 formed is wrong. Oxygen is not evolved is correct. C3 cycle, CO2 acceptor is RUBP, 18 ATP are utilized per glucose formed, correct. C3 cycle, CO2 acceptor is RUBP, 18 ATP are utilized per glucose molecule is very much correct. C4 cycle, primary carbon dioxide acceptor is RUBP, 30 ATP are utilized per glucose molecule. Here C4 cycle is correct, 30 ATP are utilized for glucose molecule is correct, but primary CO2 acceptor is not RUBP, it is phosphoenol pyruvic acid. So, identify the 2 correct items are 1 and 3. Non-cyclic electron transport, NADPH2 form, oxygen evolved is correct. Cyclic electron transport, NADH2 formed is wrong, oxygen is not evolved is, NADH2 formed is wrong, oxygen are not evolved is correct. C3 cycle, CO2 acceptor is RUBP, 18 ATP are utilized is correct. C4 cycle, primary CO2 acceptor is RUBP is wrong, it has to be PEP, but 30 ATP are utilized per glucose formation is correct. So, the correct are 1 and 3, second option. So, these are some of the important aspects we discussed in photosynthesis chapter, and I hope this will be useful for you. Almost all important aspects are touched here. In the next class we discuss respiration and nitrogen metabolism concepts. So, I wish you all success in the coming examinations.